In this video, we're going to go through the various stages of meiosis, and then we'll talk about how sexual reproduction gives rise to genetic diversity. Now, unlike mitosis, meiosis takes place in two cell divisions. Meiosis I is the first cell division, and that is what separates homologous chromosomes. So by the end of meiosis I, you'll end up with having two cells from that one cell, but each of those two cells now are haploid and no longer diploid. But the chromosomes within those two cells are duplicated, and so meiosis II separates sister chromatids. So here's a little rundown of what's going to happen here in meiosis I. So we're in interphase, and then the cell receives a signal that it's going to begin going through meiosis. We have what's called prophase I, and prophase I, there's all sorts of important things that are happening, and the most important of which is called synapsis, and that's the process by which homologous chromosomes pair up. And those pairing of homologous chromosomes are called tetrads, and we'll look more detail on those in a minute. Then we have metaphase I, where those homologous chromosomes that have paired, so those tetrads, align along the metaphase plate. And then we have anaphase I. Anaphase I is the separation not of sister chromatids, but of homologous chromosomes. Okay, so let's take a look at each one of these in a little bit more detail. Notice I'm saying prophase I and metaphase I and anaphase I and telophase I. That one refers to the fact that we're talking about meiosis I. We're talking about the first cell division of meiosis. Now, prophase I is the longest phase of meiosis. Remember, in mitosis, the longest phase is metaphase. In meiosis, the longest phase is prophase I. Because what's happening here are all of the homologous chromosomes who weren't associating with each other at all before this point now suddenly must find each other, and they come together and they form a tetrad. Why a tetrad? Tetra meaning four. 1, 2, 3, 4 sister chromatids. At this point, what's happening is some of those non-sister chromatids, right? Sister chromatids from homologous chromosomes. Non-sister chromatids are beginning to cross over each other. And the point at which they cross over is called chiasmata. And at those points, there is recombination occurring. So what we see here at synapsis, when those two, so here is a duplicated chromosome in blue with two sister chromatids. The sister chromatids are held together by these cohesion proteins. And here we have, again in red, we have sister chromatids. So it's a duplicated chromosome, and the sister chromatids are held together by those cohesion proteins. And notice there are these breaks here that occur at points called chiasmata. Next, these two homologous chromosomes, the blue and the red, pair, and that's called synapsis. And at synapsis, you have synaptonemal complexes forming. These are proteins that you could barely see in green here, and these proteins are interacting with each other such that the homologous chromosomes are sticking to one another. And notice these breaks, these double-stranded DNA breaks are occurring here at points of chiasmata, and they are going to then be repaired in such a way where blue is now going to be connected to red, and some red is going to be connected to blue. And so we get some recombination occurring, and we'll get back to that a little bit later in regards to talking about genetic diversity. Here is those repair, right? That there's those double-stranded breaks are now being repaired such that blue is now connected to red and red is now connected to blue. And so you have recombination occurring. Okay, and this crossing over at the points of chiasmata is what is going to be responsible for the recombination that we see here. And so When we go through when we finish meiosis one and we separate the two cells, now these chromosomes neither of these two chromosomes look like the parental chromosomes, right? They're recombinations of those parental chromosomes. And again, this will become important when we start talking about genetic diversity. Now, metaphase one, these tetrads, these pairs of homologous chromosomes line up at the metaphase plate. During anaphase, these pairs of homologous chromosomes separate. So notice we're not separating sister chromatids. The sister chromatids and thus the centromeres are still intact. Okay, that is a big difference between anaphase 1 and anaphase of mitosis. Then we have telophase 1, which now the cleavage furrow starts occurring, so the two cells are going to separate. We have the nuclear envelope starting to form. Now let's take a look. Let's answer these questions. How many chromosomes are in each cell? Well, we've got 1, 2, 3 chromosomes in this one, 1, 2, 3 chromosomes in this one here. How many sister chromatids are in each cell? 1, 2, 3, 4, 5, 6. So what is the state of these chromosomes? Well, in this cell and in this cell, we're now at haploid, because if you look at any given chromosome, you won't find its partner because its partner is in the other cell. Okay, so these are haploid. And then finally, the important answer to this question here is each one of these chromosomes that consists of sister chromatids means that they are duplicated chromosomes, which means those sister chromatids need to be separated. And that is the job of meiosis II. In meiosis II, each of these two cells that came about from meiosis I, that consists of haploid, right, the haploid set of chromosomes, the chromosomes that are duplicated need to be separated. Those sister chromatids need to be separated. So what's going to happen here, for each of these cells, we're going to go into a metaphase 2. So here's a prophase 2. Notice we don't have any pairing of homologous chromosomes because homologous chromosomes don't exist in this cell. It's haploid. Then we go to metaphase 2, and now we don't have tetrads aligning along the metaphase plate. We just have duplicated chromosomes. And then we have anaphase 2. We don't have homologous chromosomes separating anymore. We have sister chromatids separating. And then we have telophase and cytokinesis, okay, separating into two cells. Notice meiosis II is 100% identical to mitosis. Okay, we have a separation of sister chromatids. So meiosis II, there's really not a lot of detail to talk about because, like I said, if you understand mitosis, you understand what's happening in meiosis II. We have prophase, which is the chromosomes are beginning to condense. Then we have metaphase. They are as condensed as they're going to be, and now they line up on the metaphase plate. And each one of those sister chromatids is going to be connected to a kinetochore microtubule. Then we have anaphase, in which the sister chromatids are going to separate and move towards opposite poles of the cell. And then we have telophase 2 and cytokinesis. Now, I think I failed to mention anaphase 2, metaphase 2, prophase 2. It's important that when you're talking about meiosis, you always have a Roman numeral after the stage that you're talking about. Otherwise, it means you're talking about mitosis. And then telophase 2 and cytokinesis ends up with now a total of 4 cells from the one that we started with. So if we were to compare meiosis and mitosis, let's summarize some of the things that we've already said. In mitosis, number 1, you only have 1 cell division, right? So you start off with 1 cell and you end up with 2. In addition, if you start off with 1 cell that has 1, 2, 3, 4, 5, 6 chromosomes, each of the 2 cells will have 1, 2, 3, 4, 5, 6 chromosomes. Meiosis, here we start off with 1 cell, and because we have meiosis 1 and meiosis 2, so 2 cell divisions, by the end of it, we're going to end up with 1, 2, 3, 4 cells. In addition, we started off with 1, 2, 3, 4, 5, 6 chromosomes, And each of these 4 cells only has 1, 2, 3, 1, 2, 3, 1, 2, 3 chromosomes. So those are some pretty big differences. In mitosis, the homologous chromosomes, like these two large, the large red, the large blue, do not associate with each other at all. They don't form tetrads. And so you have just a lining up of every single chromosome along the metaphase plate, and then what separates are the sister chromatids. In meiosis 1, that doesn't occur. In meiosis 1, what you get is in prophase 1, you get a pairing of homologous chromosomes forming tetrads, and it's the tetrads that line up along the metaphase plate. And it's homologous chromosomes that separate into different cells, not sister chromatids during meiosis 1, or specifically anaphase 1. It's not until meiosis 2 in which the sister chromatids separate. So this is a fantastic figure to compare and contrast meiosis and mitosis. Here is another summary of the difference between the two. And I'm not going to go through this. You can pause here and study it on your own. Now, meiosis provides 3 sources of genetic variation. So why is it that the offspring that we see from 2 parents don't look identical to either of the parents? Or why don't they look identical to their siblings? And the first, and we're going to come back to this when we talk about Mendelian genetics, is called independent assortment. Now, independent assortment means that when a tetrad lines up on the metaphase plate, it does so in a way that is completely independent of the way all the other tetrads are going to line up on the metaphase plate. Which means that one possibility of the way in which these tetrads line up is like this, where we have the blue chromosome on the left, so the one derived from mom, and the red chromosome on the right, which the chromosomes derived from dad, let's say. And then when they separate, then you end up with gametes, sperm or eggs that look like this. One blue, big blue, little blue. Big red chromosome, little red chromosome, big red chromosome. So with all the genes that come on those chromosomes, we have a selection of gametes that look like that. Now, remember, because of independent assortment, these tetrads are going to assort on the metaphase plate independently of one another, which means if this guy here comes together, this tetrad where the blue is on the left and the red is on the right, it is completely random as to how the other tetrads are going to align. So it's just as likely that this one here will line up red on the left, blue on the right, instead of what we had in possibility 1 here. If they line up like that, look what happens now. Now we have gametes, we have sperm or eggs that are genetically different than the sperm and eggs in possibility 1 here. And that is the exact, these are sperm and eggs that are being produced by the exact same individual. So when this person, when their germ cells, the cells that are destined to give rise to sperm or eggs, when those cells go through spermatogenesis or oogenesis, okay, the production of sperm and eggs, some of their sperm and eggs may end up looking like this, and some of their sperm and eggs may end up looking like this. It just depends upon independent assortment. It depends upon how those tetrads are going to line up during metaphase 1 of meiosis. The next form of genetic diversity is one we alluded to much earlier in this little talk here, is that when homologous chromosomes pair during prophase 1, when synapsis occurs, you get crossing over at the sites of chiasmata, you get recombination occurring, and now you get little pieces of a chromosome that used to be on the red chromosome is now on the blue chromosome, and vice versa. So that means that each one of these chromosomes is going to be genetically unique because of this crossing over. And finally, random fertilization. A female will produce eggs with 8.4 million different combinations of chromosomes, and that's as a result of independent assortment and the fact that humans have 23 pairs of chromosomes, or 46 chromosomes. A male will produce a sperm with 8.4 million different combinations as well. So when one of those random sperm fertilizes one of those random eggs, the possible combinations of zygotes are 8.4 million times 8.4 million equals 70 trillion combinations possible. So a man and a woman have 70 trillion possible genetically unique offspring. This is why you and your siblings never look identical, unless you're an identical twin, of course. Okay, so we will come back, particularly we'll come back to this idea of independent assortment and even crossing over in the next couple lectures. But let's take a moment now and look at some aberrations that may occur, or alterations that may occur that are not normal, right? That are not intended to occur, and these alterations in chromosome number are a result of something called nondisjunction. And now you can have nondisjunction in meiosis I, and you can have nondisjunction in meiosis II. Either way, you end up with gametes, sperm and eggs, with an unusual number of chromosomes. So let's take a look at here. We have 1, 2, 3, 4 chromosomes in this diploid cell. Therefore, we're looking at 2n equals 4. Now, during meiosis I, the homologous chromosomes are supposed to separate into 2 separate cells. But because of nondisjunction, sometimes what happens is one of the homologous chromosomes doesn't properly separate. It goes with the other one. Which means we leave one of the cells, at meiosis II, we leave one of the cells that's missing a chromosome, and we have another cell that has an extra copy. So we have 1, 2, 3 chromosomes, instead we're only supposed to have 2. Which means the gametes here that are formed, we have an n-1, so we're missing a chromosome, and we have in these gametes an m+1, where we have an extra chromosome. Now, when one of those one of those gametes that has an extra chromosome is fertilized by a normal sperm, for example, we now have an offspring that has 1 extra chromosome. So we say 2n plus 1, meaning she or he's got a full complement of human chromosomes, but they have 1 extra 1, and this is called trisomy, or 2n plus 1. This long chromosome, instead of having just 2 copies of it, this person has 3 copies. Or if the egg was fertilized by a sperm and the egg was missing the chromosome, right? If it was one of this egg right here and is fertilized by sperm, then what we have is this chromosome here is only going to be represented 1 time when it's supposed to be represented twice. That's 2n-1. That's called monosomy. When a person an individual has 1 extra copy or 1 less copy of 1 chromosome, we call that aneuploidy. And there's one very common disease that entails aneuploidy, and that is called trisomy 21, or 3 copies of chromosome 21. And these individuals have a certain set of symptoms and characteristics because they have 3 copies. Whatever genes lie on chromosome 21, they have 3 copies instead of 2 copies. And that genetic imbalance causes these various phenotypes, okay? Now, most aneuploidies are lethal in humans. You'll never see a human with trisomy 1. Right? It's no coincidence that chromosome 1 is the largest chromosome, has tons and tons of genes. So if you have an extra copy of all of those genes, that's going to be lethal. That's going to be embryonic lethal. Polyploidy is when you have a complete set of chromosomes that have been duplicated. So instead of having just 2 sets of chromosomes, like a diploid organism, you have maybe 3 sets or even 4 sets. There are some organisms that exist this way. So this Plains rat is tetraploid. It's got 4 sets of chromosomes. If you look at a single chromosome, you'll see 3 others that look like it. Many of our harvested or agricultural products, like strawberries, for example, are extremely polyploidy. These strawberries here are octoploid. If you look at the wild version of those strawberries, that won't be the case. They'll be more diploid. All right. With the understanding of meiosis, you can now start to understand Mendelian genetics. And if you are sketchy on meiosis, go back and look at it over and over and over again, because understanding Mendelian genetics is absolutely dependent on understanding meiosis. And we're going to be sort of tying the two together during our discussion of Mendelian genetics.